open the session with Simon Eickhoff. Simon is an MD by training who did his PhD in neuroanatomy before moving to cognitive neuroscience, MR imaging, and finally machine learning. He's the director of the Institute of Systems Neuroscience at the HHU Düsseldorf and the director of Brain and Behavior Institute at Jülich.
Thank you. Good morning. Thank you for putting together this really interesting symposium. And I will start by showing a positive outlook before then dialing back a bit. So let's go. Oh, okay. Oop. Now we go. So clinical application, that's basically the search for biomarkers. And if you look at the current literature, there are hundreds, thousands, 10,000s of papers that describe biomarkers for all kinds of neurological and psychiatric disorders. In that context, however, it is important to realize that classical biomarker studies are always based on within sample comparisons or associations. So as an awful reminder maybe to early statistics classes, really what you do is you have your group of patients and you have your group of controls, and then all you are asking is the difference in mean between these two groups surprisingly big, so surprising that you wouldn't consider them coming from the same group. Now, the problem is that these kind of biomarkers may be somewhat misleading. So I'll show you an example here. That's a highly significant difference, p less than 0.001. You see, look at the standard errors. I mean, isn't that a good biomarker? Didn't you find something about the disease that is really applicable to clinical use at some point? Well, let me show you the very same data, just in a somewhat more honest fashion. Okay, who thinks that now that this is a good biomarker? So the critical thing is that biomarkers from classical statistics always pertain to the group means. On average, patients have more or less or whatever, but individual statements are usually not possible. This is now changing in the context of machine learning becoming more common and more prevalent. Now, what's the key point here? And I'm not talking about deep learning, XGBoost, and all the architectures, whatever. The key conceptual point is that you're not testing for group means, but rather you train a model on the available data on your training set that tries to find a multivariate relation between the input features, let's say the brain imaging data, the connectome, and an outcome, a target, for example, disease classification, symptom scores, and so on. And then you go to new data, a previously unseen test group, and see whether that model actually generalizes. So the key point is that we are not, as in classical statistics, look for a description of the sample we have, but for the generalization to new cases. And if we think about clinical application, this is an absolute must, because you're not interested in the group of depressive patients that we analyzed in 2007. You're interested in that one person that's now sitting in front of you. And so, with all the large data sets that we have, all the methods, the high-performance compute, we are ready to go, aren't we? Well, and this is sort of where the positive outlook is going to be a little bit clouded over the next couple of slides. So let's have a somewhat closer look at the machine learning pathway. And over the remainder of the talk, I'll just give you a few highlights, snapshots of things you may want to consider before you're too enthusiastically believing that you found the next big thing. So what you start with is your data, for example, a connectome and a target measure, for example, a disease label or a symptom score. And then you always have to first extract the features from your data. So that's a critical thing in neuroimaging. We usually don't use the raw data, but we extract some form of features from that data that then, together with the target, are used to train the model. Then we validate it in new subjects, as I described. And finally, we are hopefully ready to have the product for deployment. But as I said, there are quite a few potential road bumps along the way. And that starts already with the actual data. Sticking with connectivity, that's actually a quite interesting study that we did quite a few years ago that seems to be rediscovered over and over. And the thing is, if you want to, for example, extract resting state connectomes, there is no such, let's say, natural thing as the connectome in your data. But rather, you have to go through quite a few pre-processing steps. There is things like denoising, confound removal, global signal removal, and so on and so on. And what's actually quite interesting, and particularly interesting that I saw a paper coming out just last week that basically, again, found the same thing, is that if you do remove more of the nuisance signal, if you do global signal regression, if you do a more aggressive confound cleaning, reliability of your data decreases. So if you have cleaner signal, you have less reliable signal. That may or may not be what you're after in the clinical setting. But even if you do have a good signal, then you still need to represent that signal in a form that you can later then put it into a learning algorithm. And if you think about brain imaging, if you think about connectome analysis, we can't do a voxel-wise connectome in any meaningful fashion. Not only is that incredible to handle in terms of the size of the data, but you also would have a horrendous feature-to-sample ratio. So there's no data set where you would have more subjects than you would have features from the full brain voxel-wise connectome. So you really want to do something like an atlas-based compression. I guess everybody who's ever extracted a connectome is doing that almost automatically. You use the XYZ atlas and then extract the connectome. Well, the XYZ atlas is actually already somewhat of a problem because there are quite a few different atlases available, and all of them are biologically plausible, are well constructed, and are used very, very frequently. Does it matter? Well, as you see here, by a study by a former PhD student, Justin Domhoff, we extracted resting state connectomes and did this for a couple of different atlases and just computed standard graph matrices. And without going into the details here, I hope you immediately see that depending on the same data, what kind of atlas you use for compression, your actual features and their graph descriptions look substantially different from each other. So choice of preprocessing and choice of atlas compression basically means that there is no such thing as the connectome you give a learning algorithm, but you can basically give in almost whatever you want depending on how you process the data up to here. Now, let's have a brief look at the targets, because again, this is something we just take for granted. But what's actually quite interesting is that the effective outcome of your learning models will massively depend on the target. I really recommend this preprint by Martin Gell, almost former PhD student, who actually looked at machine learning, connectome-based prediction of, in this case, age. It's a very simple, reliable measure. And then he artificially degraded the reliability of the target. Same pipeline, same data, just adding some noise to the target, to the age scores, and basically pretending that you're doing a less reliable target. And see what happens to the prediction accuracy. It basically craters. Now, is that something that we also find empirical support for? Yes, as you can see here, for example, in the UK Biobank, reliability of the test-retest reliability of the targets is directly correlated with prediction accuracy, and the same thing we also find in the ABCD study. So at this point, if you're still semi-motivated to go forward, we know that the preprocessing matters, we know that the compression and the representation matters, and hopefully you all take away that the targets massively matter. So if you have an unreliable target feature, no sample size, no most fancy learning algorithm can ever save you. And this has a lot of implications for clinical classification studies, where we know that the targets are at times a little bit shaky, but in particular also for continuous predictions. So as a rough measure, the more reliable, the better, and it's usually compound scores that are more reliable than individual scores. Okay, now let's finally go to machine learning, and maybe things get a bit better there. Well, as you can see here, this is a study by Amir Omedvarnia, a postdoc in our lab that's now just out in communications biology. We looked at predicting individual cognitive phenotypes, let's say here cognitive speed, from resting state fMRI. And what are all these different lines? These are all the hype cool stuff you can do with resting state data. All the time series metrics, connectomes, all the fancy analysis techniques. And what's very nice is that we can see whether we can significantly predict cognitive speed, and also very nice, if we increase the size of the training set, prediction accuracy gets better. That's good. What we didn't really see is a substantial difference between the different methods, which was a bit, let's say, uncomfortable. We actually hoped that things like entropy and different dynamics would actually be more effective, but in the end, it was not. Well, the bummer comes here. This is a prediction only based on age, sex, and intracranial volume, not using any connectome data at all. Oops. What I'm getting after here is that we must absolutely be careful. We want brain imaging to work. This is what we do. We want connectome prediction to be the next big thing. But I think this is an absolutely critical control analysis. How good can you actually predict your targets just by looking at confounding factors such as socio-demographic or other influences? And we're doing this now on a regular basis, and it's humbling, I have to state. Just another example of the same problem. How good can machine learning differentiate male and female brains? Really well, as you can see here. Basically, something no radiologist can do, tell whether an MRI is male or female. And with just about 1,300 training subjects, we could get a very nice classifier. Up to the point where we saw that basically all the algorithm did was looking at brain size. So basically, correctly classified females were the smaller ones, incorrectly classified females were the taller ones with the larger heads, and for the males, it was the other way around. We did find some ways to correct for that, but the take home here is machine learning algorithms just love shortcuts. Okay, and that gets me to the final statement I want to do, which is that human variability is likely low dimensional. Again, a paper I really want to highlight, Vera Kormeyer here. It's a preprint at the moment talking in detail about confounds and the conceptual aspects of this. So basically, what we propose is that confounds really have to be seen as a gradient. There are things that are just stupid, bad design. But then there are also things that are likely implausible but could have biological links. My favorite there is a UK Biobank GWAS hit for the question, how often do you eat bacon for breakfast? See, it sounds odd, but maybe there is something on saline receptors and so on. And then you move on, and this is where it gets really tricky. Potentially your confounds and your targets may be reflections of some underlying latent dimension of human variability, maybe driven by a common factor, or in the end are just measurements of the same biology. So, in the end, I would argue everything is a confound. And there is no such thing as a clearly deconfounded analysis. Critically, this does not only have this statistical dimension, how strongly is the confound correlated with your target, but also has a conceptual dimension. So here we actually almost dwell a bit into the realm of causality. How much is a confound actually a statistical confound, something you want to get rid of, or something that is truly inherent to what you actually want to look at? Why is that so critical when we think about machine learning and clinical applications? The key point here is what's called confound drift or confound shift. So if you train a model on a certain population, and that model does take a little bit of a shortcut through confounding factors. And remember, I said that earlier, every model wants to take shortcuts. And then you apply the model to a different population where the relationship between target and confound may be different, or the confounding factors may be differentially expressed. And this has been shown very nicely by Jingwei Li, who used connectome-based prediction, predicting cognitive phenotypes in some of the large US data sets like HCP, and then basically looked at prediction accuracy in white and African Americans. And basically what she found is that if you train a model on the majority, then it does all sort of bad things on the minority. You can have lower accuracy, you can have offsets, you can have slope changes, and so on and so on. Just some examples. And this is basically explainable through the fact that we have differences in the confounding structure. Last but not least, a shout out to look beyond your expertise, that may be imaging, that may be machine learning. When we want to move towards clinical application, it becomes critical to also look at what's called the ELSA, ethical, legal, and societal challenges. And basically all the points that I've mentioned up to now really accumulate in acceptance, accountability, and in the end, usefulness of the algorithms. And with that, I would like to close with two images from William Turner that I think represent our state quite well. There's a lot of full steam ahead enthusiasm, so we can actually do a lot of things that we could only have dreamt of a couple of years ago, but we need to be careful because these complex workflows are susceptible to many influences, and conceptual and biological challenges, such as multi-confounding, will immediately lead to ethical, legal, and societal implications. Thanks a lot.
This question, we're open for questions now. We have one in the app already. Can't the choices of Atlas preprocessing or confounders be optimized as hyperparameters in the machine learning pipeline?
In theory, yes, but I would kind of advocate for caution by using a purely data-driven view on the field. And this is particularly true for the confounding, because in the end, these confounders are not so much a statistical problem that you can solve with statistical methods, but they are a conceptual problem. Do you want to remove something or not? And what does it mean to remove it? So as one example, if you look at anything that is physically related, then basically, let's say hand grip strength, walking speed, and so on. If you use sex as a confounder, you're basically removing testosterone effects, and this is directly and causally linked to muscle strength. You want to remove that? Fine, but you should know why you remove it and what that means. And that's something you cannot solve with a hyperparameter tuning. That's actually thinking.
Okay, then one last question, quick question from the audience. So
it works, yeah. You mentioned a lot of problems now, a lot of challenges. I would be curious to hear from you, what is the future roadmap? What are the solutions for all these problems? How do we get reliable biomarkers? And maybe you could summarize that very briefly in a few points.
Yeah, well, I still think the future is bright, but I am warning against a yo-hey enthusiasm at the moment. In my view, nothing is ready yet to actually be used in a practical setting, in a hospital, in a court, or whatever. But rather, I would really argue for take a step back. It's not just chasing accuracies. The goal is not to increase accuracy by 1% over the last paper. And this is what I'm seeing in neuroimaging. That's what I'm seeing in the hard machine learning field. It's always beating state of the art by at least 1% or 2%. No, that is not going to get us anywhere because we are just exploiting more and more problems. We actually have to take a step back and try to think and actually develop algorithms that are more useful, more meaningful than just being 1% better in accuracy.
Thanks a lot. I'm sorry, but we have to move forward, but great answer.